No, I didn't know that, Chris.
It's because they have Peneus envy. I guess you didn't get it. Peneus? Like the genus?
Yeah, okay.
Oh.
We're
okay,
gonna get in trouble.
You want
to see this?
Invertebrate zoology humor.
Please, that thing.
I'll be here all week.
Yeah, there's also a sponge on the lower left that is different.
ROV nav bridge.
When you zoom out, you'll see it.
Okay.
Let's see. It's on the side of there. It is that yellow thing in the bottom of the boulder.
Oh, over there.
Yeah.
Let's see. Go ahead, video.
Oh, that's a weird-looking thing.
Yeah, so this could be my buddy that we collected yesterday, this yellow Poliopogon species. And it's in a very weird location where it is, but you see, you just don't get members of this genus with this yellow color to it. And it looks like it was damaged. You see this upper
Yeah.
right here?
Yeah, I'm starting to wonder if there's just something here that's taking a bite out of these things. Maybe it's that grenadier.
Well, you don't see these sponges that are predated on very often. They've got some serious defenses in the form of all their spicules. What's that?
Oh, looks like there's a cloud of rise on the right.
The focus here.
Yeah, this looks like a, well, if this is a cloud-arised sponge, then conceivably those thin projections off to the side are what they use to capture small crustaceans.
Actually, did we get that small one? Do you want to zoom back in one more time?
No, we got it.
Okay.
Oh, that's fine.
I'm good with it.
Yeah.
I'm looking 320, and that's where we're headed.
Headed 320.
So things are looking up, even though we've seen that sea cucumber before. There's more of them. We're seeing them more frequently. So that may indicate that there's more food, and thus more productivity, and thus more animals.
Something's right over here.
Some nice pillow flows here. These big sort of bulbous
I know,
rocks.
so lovely. Sometimes it feels like I could just be jumping along those right now.
Is that an anemone at the base of that sponge?
Yeah.
Oh,
it
no,
is.
it's on the rock. There
Yeah.
it is. Another one of these big old things that we call Actinostolidae.
Here's a spiral something on the right side there.
On the right. Did you want to see this up on the... Okay. Where do we want to see first?
Let's look at the coral first. Sort of the one that's...
You mean the anemone or the coral?
No, there's a spiral coral on the right side there.
On the right side?
Yeah.
Well, how about I
Edge of the
snap zoom on
screen.
the anemone since it's here?
Okay, let's just
sorry.
snap zoom.
Yeah, sorry to sound back and forth.
You just
Yeah, I don't know if this is a characteristic, but you see how there's those lineations on the disc. Those were the same lineations we saw in that small one that was withdrawn, so I think you're right. It's the same thing.
Yeah, and we did think that a few years ago until somebody straightened us out. We were actually calling these anemones Exosalactus, and then we got a different opinion and convinced us that it wasn't.
So they said there was a coral?
Yeah,
on
to
the
the
right.
right, there's a spiral sort of.
Oh, back in there.
Yeah.
Okay, sure. Let me go back there. So we'll just pass on these guys right now.
Mashkor, what is the matter with your seamount? There's not very many animals on this thing. What's the story? Yesterday you did so well on that other seamount. Yeah, we did. Look at it. It's all rocky right here. Yeah, well, we're getting there. But, you know, I don't know. I mean, Mashkour Malik has joined us in the control room for a few minutes, and he is the person who runs the team that did the marvelous mapping of the seamount last night and early this morning.
Try to get to the tip here.
So you mentioned what this is, right, Chris?
This is some kind of octocoral, I think.
Yeah,
but
it's Iridogorgia.
It is Iridogorgia. It looks really beaten down for.
Yeah, it's not great, is it? It's a little
I didn't
ratty
recognize it. I guess that could mean that there's a starfish nearby.
Hello, co-pilot.
Good afternoon.
Just to let you know, you are currently in the middle of a move, 30-meter move at 320.
320, copy that. And the ship is still moving?
We should be coming to stop soon.
Soon, and we'll go for another 4 or 5 minutes then.
Copy that.
Copy that. Thank you. Also,
RV nav bridge move
previous co-pilot wasn't sure if there was something in...
Copy bridge. Go ahead.
He wasn't sure if there was something in the port rock box.
But a bamboo.
So there is both
Any interest?
rock boxes are full.
Let's take a peek at this.
We have something in the port inner bio box,
right,
which might float out. So
okay, so don't open the port at all.
Your dinner should
Copy that.
be totally burnt
And
by
how
now,
about
or you
starboard is
consumed
empty?
it.
that is
If you're
correct.
near the
No
phone
biosamples
and
on
near your
starboard
video
bio
or
boxes.
zooming in on
Copy
another
that, thank you.
bamboo that's got these extraordinarily long needles. Okay.
This may be what we were imaging earlier, and what's really informative is that one that we saw earlier did appear to have branches, and this one is not, and so this raises one of, as you know, my favorite things to talk about, and that is the difficulty of discerning these species
slowly to the base.
Just based on their colony growth form, because you don't always know what stage of life you're seeing it, and whether what you're looking at is a branching form or an unbranched form. And the way the current taxonomy is set up, anything that doesn't have a branch and would be in the subfamily of bamboo corals would be put in the genus Lepidices. But as you've heard less than I say
pilot, we are
times,
looking down and maybe a little back at you.
Taxa, we know that there's lots of different ways to build a colony, and there are other things you have to look at besides simply the way they branch.
Yeah, thank you, Scott. And I think you left it at J clade. Is that correct? Or I think you were toying with S clade or I1 clade. I'm trying to remember now.
Yeah, sometimes I never know either. I did leave it at J clade before, and I'm not entirely satisfied with that, but I think that is the way we should leave it for the moment.
Okay. Thank you, Scott.
In fact, I will go further to say that in my database, I'm looking at images from 2015 when we collected one of these J-clade unbranched whips with these very, very prominent spines, and it looks a lot like what we were just looking at. And it's why I hesitated before, because it was the branched one. So nice to see the unbranched one.
Hey, pilot, I'm going to turn my head to keep you in view.
Copy.
You need to come back to
we're coming
port
up on a
when
nice
you get
glass
underway
bunch.
again.
Well, let's take a look because there's something on it. This is one we've seen earlier today, but it might have some commensals on it. Right on those white things.
Oh, yeah. Yeah.
Okay, come on in video.
The snails. Yeah.
Oh, that's an interesting one.
Wow.
Huh. Interesting.
Tilt down a little bit.
Slightly.
Maybe I've seen snails on these sponges before, but I can't remember.
That might be too far
Are
there.
they hermit crabs?
No, there's the.
Oh, that's
the
a hermit.
tentacles.
Like
No,
this
that's
in
a
a
hermit
video.
crab.
Are you
That's
sure?
good.
Yeah, see the leg coming out on the side.
I don't know. Those look like tentacles, and then those look like the eyes of a snail. Well, at the very least, I mean, like, does that one looks like a snail, right? Because I think they're attached with the proboscis. I mean, that one might be a hermit crab, but the other ones look. Let's see. Are there any others in view? Maybe we're missing something.
You want to look around for other ones?
Yeah.
Okay. Zoom out
Well, there's
slightly.
one to the
There's
left.
one up top here.
Yeah, up top here.
You want to see
just
that
get
one
a little better look at it. Well, I think that's the snail. That doesn't look like a hermit crab to me.
Yeah. Well, so at the very least, there are snails here.
There's one on the top left there. Do you want to see that one?
Doesn't seem like it shows any better.
Here.
We'll try video, see if I can do a stable shot here. With tilting up.
Smack zoom.
Copy.
Was that Caulophacus?
Something red on the top right. Something right at the top.
Sure. So sponges, especially glass sponges, are what one calls ecosystem engineers. Their physical structure provides habitat for different types of animals.
Blown out wide.
We've often seen a multitude of different animal phyla present on different glass sponges at depth, and we can see a perfect example of that here. This one has, for example, 1, 2, 3, 4, 5 small snails living right on the surface. But if we were to turn it around or look around at some of the other glass sponges, we'd probably see brittle stars. Sometimes little crustaceans live in there. I'd almost bet that we'll probably find some if we were to look through it. But sponges are an important component of any ecosystem because they literally are an ecosystem for a bunch of other smaller animals. How's that?
Lasers, copy that.
Okay, we can move on. Thank you.
You're wide. Your mic is not open.
Watch these. We're just going to get quick lasers on here and switch out pilots.
Lovely. Thank you.
Roger that. Thank you.
Is our light out here on the panel? Yep.
So we're having a bit of a pilot change in the front row here, so stand by. It'll just be a minute.
Welcome aboard, pilots.
Howdy, howdy.
Howdy. I don't know if Andy told you our heading was going to be 320, so I turned to keep him in view, so you need to go to port and push out. Yeah.
All right, copy that. Sorry, what was that, Chris?
Oh, nothing. Just saying, how's it going, guys?
No, it's going great.
Good.
Where is everything?
Oh, well, that's a good question.
It's that
last
way.
time I was piloting, I mean, you couldn't even sit down.
There's stuff over that way, whichever that way is.
I'm assuming we've seen this glass sponge.
Yes, sir.
All right.
I think moving upwards and seeing what's on these higher platforms is of interest.
Roger that.
Thank you.
Ooh, our motor installation's way down. What's going on? Now it's back up. Strange. Can you note that in the log?
Yep.
All right, Don, if you can pull up a little bit.
Okay.
I'm pretty close to you right now. Yeah, it's down to 7,700 km.
Josh, I don't know if they relayed to you, but in general they wanted to make tracks.
Yeah, copy that.
There's this dark
Yeah, come on into you.
blob. Whoa, what is that? It's some kind of slug.
It's a pluribranch. But
It is a
very
pluribranch.
unusual one.
Whoa, it's 10 centimeters across.
It's going to the edge.
Chris, do you know which one this is?
No, and actually I would offer this as a possible collection option here, because these are really rare, and this is so different than any I've ever seen. And it's large enough, I think we have a chance.
Wow.
Pilot, what do you think about this? Is there any chance we could collect this?
We can get a shot.
I'll throw it out to the group here.
Let's get some good imagery first.
If you are getting caught up with us right now, we have a really strange pluribranch, but
Pilot, what was that number that you saw
Chris
earlier
I've never seen
on
it
the
before, but
insulation
maybe
resistance?
one of you has.
So the motor insulation resistance is
Was 17?
dropping down.
Oh, yeah, 13.6.
Yeah, periodically.
Yeah, but yes from Scott France.
Which is a bit concerning. Wow, that thing can really move.
And he's about to drop off.
The color also suggested yes. Yeah, we got a consensus. Everybody is giving a very
There was
enthusiastic
something
yes.
else up on that rock to the left as well.
Yeah,
It's dropping again, 77.
There's a red
yeah,
sort
I
of
saw
circular
red
thing.
something or other. But
Yeah.
whoa, that is different. Holy moly, yeah. What is that red thing?
Look how big it is.
Yeah,
It's like nothing, 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 and then boom,
he's gonna hit over
giant
that
monster
ledge pretty
Cthulhu
soon.
pluribranch.
All right, what do we have in bio boxes?
We've got something in the left bio box. We prefer not to open that one.
All right, copy that.
So,
All right, can we ramp up hydraulics?
copy that.
Yeah, port inner is the only one, but it's a floater.
All right, so we're going to go starboard.
Yes.
This is amazing.
Nothing in starboard other than the rock box. Nothing in starboard by.
All right, positioning is going to be important on this one. Let's see. Let's do a zoom on him. Let's get some good imagery before I go in.
So while we're sort of looking at it, wow, that thing is nutty.
Actually, let's
gorgeous.
come out because he's going to crawl under.
He's gonna
that
crawl
rock.
under. That's what I was worried about.
I was going to say, what's that red thing on the rock to the left? But
Yeah, let's get this guy in the box, and we'll look at that
Yeah.
red thing.
All right, let's get that box ready.
Copy that.
It's pretty rare to see sea slugs down here. They're
yeah,
not
I
a
know,
common
it's really
animal at all.
strange.
Well, any sea slug, even the
Yeah.
other more familiar
Yeah, I mean, I thought that they all pretty much just pinch out after, I don't know, about 1,000 meters or so.
Can I help you with any zooms?
We're good for right now. Don, if you want to bring up
Mini Zeus.
yeah.
Copy that.
Wow.
That's experience for you.
Grab mid-water. There you go. Nice catch.
Kids, don't try this at home. Actually, they could if they could build a manipulator arm and try it on little kids like these.
Now he's out of reach. Alright, I gotta reposition and go for him again. He's not escaped yet.
I'm going to tip down on mini Zeus.
I don't need it.
Nope. Okay.
Let's open the drawer.
All right. Okay. Coming out. It's all the way.
All right, now you can tip down on mini Zeus if you want.
Okay, drawer closed.
He's a little damaged, but he's in there.
You got him. Thanks very much.
Wow, thank you.
Hey, video.
We saw another deep sea.
Is there any way you're able to pump the hydrophone through my headset alone?
Earlier on in the expedition, a few days ago, Terry Gosliner gave me a name.
If not, don't worry.
Wait a second. Pilot, before we leave here, there was a red thing on the opposite side of the little boulder there once you stow the arm. That I wonder if we could swing around and look at.
Yeah, absolutely.
Thank you. And good job.
Thank you. Whew. All right.
Hydraulics off.
All right, I'm done
that's
with,
copy. Is there any way we can record the sound somewhere else right now?
Camera.
Nope. Okay.
There he is, the red thing.
Thanks. Can we record this video?
Come on in video?
Did you say come in or
yes,
live?
Come on in.
What is that? Oh, it's an Anthomastus. It's closed up.
So you got something?
Yeah,
is he eating something?
I've seen them many times.
That's noteworthy. It might, maybe that's what the side gill slug, what the slug was eating. This is actually a good sign as far as the region, though, because if there's a slug like that moving around, maybe it means there's food for it to eat. So we're probably, I bet we're just above a huge field of things.
I hope you're right. But it's definitely getting better. It's pretty dry in the beginning, but we're seeing some corals and sponges, live ones.
Did that go in starboard inner?
Yes. the waypoint.
Copy that. Yeah, I got it down.
There might be a
survey to nav.
something to the left in the water column. I thought it might be a fish. Oh, shrimp maybe. What is
Hey,
that?
Roland says you can record this hydrophone thing that I'm saying right now.
Oh, Kedognath. Okay.
Oh, that's great. Thank you so much.
Not used to them being so rigorous.
Let's get the next 5 minutes.
Oh wow,
at least.
Nice shot of.
And can you get anything previous or no? Okay, from here on for 5 minutes, please. Thanks.
Wow. Sort of see the teeth on the side and everything. These are one of those animals that if they were as big as we were, we would be terrified of them.
Inside your lens,
they can see
focus.
in a fairly wide range around them. They have giant teeth that can swallow animals that are the size of their own body.
The other one was closed.
Go ahead and roll.
Do a look at this. These are mushroom corals. So it's got probably on the order of somewhere between 15 plus individual polyps that are coming out of a ball-shaped structure. Is it? Let's see where.
Oh, I think I think I see what Roland's talking about. But, that's one of the actual.
So if we average
arms of the polyp
0.08 for
putting
the
food
rest
into
of the dive, we can get to the very end.
You're talking about that sort of top polyp with the red.
So I can put a move in as soon as you guys are ready for one.
That's actually it's actually caught something and it's sort of moving something into the.
Yeah.
Into
You looking at 320? You happy with that?
320 looks good. Yes.
Copy that. I'm going to give you a 30-meter move. Bridge, RV nav.
It's the bridge. Go ahead.
I'd like to put in a ship move, please.
Okay, go ahead.
Range 30 meters, bearing 320 degrees, 0.2 knots.
Range 30 meters,
There's a lot of lava
bearing
coming down
320
from the
degrees,
peak at some
speed
point millions of years
0.2.
ago.
Good capping.
Coming up.
Let's take a look at this little white circle to the left.
You can have that.
It's on the rocks.
Come on in.
Weird.
Maxim.
Sponge?
ROV nav, move initiated.
Copy.
Are those polyps? I don't know what we're looking at. Oh,
Yeah.
It's,
Could be a sea slug?
well, yeah. I was wondering if those are the gills coming off
Yeah.
of it. A weird.
Oh, that'd be
Try to
weird.
get
Find
in
two
close.
sea slugs at
Yeah, I'm doing that. Roland.
2,300
Okay.
meters all of a
Let's go
sudden.
pull out for you.
I don't know.
So there's a snail. Is that a snail down there to the left of it, or to the left?
Is it just a dead stock that we're looking at?
Yeah, probably a dead stock. We can move on. But there's a snail
Bridge,
down to
RV
the
nav.
left there. Oh, you know what?
This is the bridge. Go
I think
ahead.
that's
Hey,
Daza.
quick question for you. Is that bow thruster moving around pretty good right now?
This little shiny or
Or just a second ago?
Looking at right there.
It seems about normal. I don't see it right now. I
Gaza's
wasn't paying any attention just a few minutes ago, but
in 2015
well,
were
it's
bigger
spinning
than that.
up
Well,
there.
no, if
Imagine it could be a small one.
if
pretty
it
much
drops
pointing off
and
our
rolls,
starboard
then-
quarter.
Copy that. Thank you.
I'm serious. That's apparently their behavior.
Right, come on in.
Yeah, I know.
Yeah, I bet that's what it's about to do. Yeah, that's sponge stock.
Yeah.
Well, we could wait all day for that to drop and roll, but
I'm not so sure it's gauze at all. I think we're fine, pilot.
All right, come on up.
I actually measured the distance they were rolling for Mary Wickstein, who wrote that up, published the observation on it, and I looked a lot at that video footage. So if it is one, it's a really tiny one. Yes, they have shiny shells like that, some of them, but
just small white dot at the top. Seems to be a stock something
Come
another.
on in, Roland.
oh, it's a nice amphiurid brittle star.
those little forams, arborescent forams, a little cluster of them, I guess. At least that's what we think they are.
Yeah.
All right, coming up.
It's very hard consolidated substrate here.
Well, there's a fair amount of stuff living around it, though. It's not so bad.
It's quite a crack right in the middle of it as well.
Yeah.
All right, just come out.
That's another one of these anemones that's partially open, at least. I wonder we snap zoom that straight down.
Straight down right here.
I think it is.
Come on in, Roland.
Unless it's another dead. It's another. Never mind. Thank you. Wishful thinking.
Oh, sponge stocks. They're just.
It's a sponge base or coral base or something weird.
Yeah.
Oh, look at the tether. Got a loop in it.
Oh, that's a dead sponge stock.
We got a loop in it?
Yep. Check out the Titan.
Tilt down on your Zeus.
Yep. There it is.
All right. So.
Well, maybe it's not so bad if you just.
Well, can you zoom in on it? I need to do a port turn. So.
Tip. Video, can you zoom on
No,
the
you
Sirius
can
camera?
leave it. It's fine. I can see it.
Okay.
It's underneath. All right.
Yeah.
So pull up a little, Don,
Coming up.
and I'm gonna get rid of this turn. Just a second here, Chris.
No, I can see already in the co-pilot screen. No problem.
Go ahead, survey.
Josh caught a twist happening in the tether before it happened, so he's.
Yeah, could you give me maybe 2 more minutes?
A maneuver here
Thanks.
to make sure they don't get a twist in the tether. So he will be with you momentarily.
You tilt down on your Zeus.
Coming down. Going up a little bit more. It's all the back
We're
I've
getting a
got.
high-level view of what's ahead, and I'm not sure we're going to get into that incredible community today.
ROV nav, move complete.
Copy, bridge.
So now you're showing 1.7 turns. Huh?
So let's just look at the tether here.
Yep. I'm going to stretch it out. I'm going to come down a little bit.
That is quite a coating of manganese. Look at that to the left, all over the substrate.
Yeah.
I think I may have connected a lot of loose boulders there, or maybe these are funny. Well, they're lava flows, I guess.
They come down a little, so we got some more light.
Manganese-encrusted lava flows.
Looks good.
Looks good.
Yeah.
All right.
Okay.
Looks like we came down with a turn. Good spot, Don.
Thanks.
So we're at 2,200 meters right now.
This is a long shot, but there's a white ball right in the middle of that lower screen crack. Can we just take a quick zoom?
You're going to have to direct me a little bit here.
So there's a dark angular corner in the lower
you see
center.
the lasers relative to the lasers?
Down left.
Down, and then to the left.
Straight under lasers.
Yeah, right there.
Come on in.
And then more to the right. Yeah, right there. Some kind of... Oh, yeah, it's a sponge stock or something. Okay, thank you.
There's a purple there. What's the purple?
Oh, what's that? Is it sort of a pteropod shell? Oh, it doesn't matter. Yeah, that's fine. Thanks.
Okay, moving along.
Is it flattening out, or we still got some more climbing to do?
It's starting to flatten out. At least in high pack.
Look like there's something on that sponge
Let's take a
surface.
quick look at this sponge.
Come on in.
Hey, Chris.
Yeah, oh, sorry.
Oh, no, it's fine.
Oh, look at this. Wow.
This is your thing, isn't it?
Yeah, but these are different. Look at the sizes of these things. These are commensal anemones that are growing out of some kind of sponge. I'm sorry, I was addressing the chat room there because there's a discussion on how to preserve that sea slug, and we've got a bunch of people who had some recommendations. But I've got to turn my attention here to this. Whoa. It's a hard sponge. It's can't really tell if it's broken or what's going on with the top there. And then all these yellow parts are a zoanthid, looks like a zoanthid or hydrozoan, I guess, that are probably commensal on the sponge. Wow, that's a lot. And you can see some of their bodies on the other side. I can't tell if this is a tube sponge or what, or whether it's a plate. I don't suppose you could swing around and look at the other side, could you, after you're done with this?
Yeah.
This is a very different kind of commensal anemone that we've seen before. This could be something we call Cheetopleura. There's also maybe a Heteropathes black coral.
Go ahead, survey.
To the left of it.
Okay, we're good. Yes.
Do you want a partial zoom as you turn?
You can just hold this for right now. Did Fernando ever get someone from Team Electrical?
Well, if the sponge is paddle-shaped, then
For
most
what?
likely it's
So
in
the
the
motors.
About the motor?
family Incinerateridae and in the genus Tretopleura.
We're going to call
That's
around
what
to a warehouse.
We're defaulting to
Uh,
in
to-
all of these
To workshop.
paddle-shaped
Sure.
Yeah,
sponges
they
for
should
now.
be in the hangar.
Yeah, come on in.
They're not known to have commensal
Workshop,
anemones,
Nav.
which is weird about this.
Hey, yeah, we're looking for somebody from electrical. Could
Those
you
are just
come
big.
upstairs, please?
Weird, isn't it?
Commensals, yeah. those are
No,
really
just
big,
somebody
the
from
biggest I've ever
electrical,
seen
team
with
electrical.
it.
Yep. Come to the control room,
There's not
please.
as many on this side, maybe, or
You can just tell them what the problem is.
What's the problem?
The motor DC, the motor insulation resistance is fluctuating.
Okay. Hey, workshop, Nav.
He's probably on his way up. Doesn't look like a tube.
no, it certainly doesn't, does it? So I'm gonna say unsedentarity or eurydidy, one of the two, but and then with, I guess, colonial anemones or zoanthids or something.
Something.
Wow, that's a very fascinating new observation. Thank you very much for that. You?
Go workshop.
Oh,
The motor.
yeah.
I
saw
don't
a
know how
bunch
to
of
plot it.
small things on the bottom there.
You might want to come up and show us how.
At the base of that sponge we were just looking at, there's a whole bunch of small, tiny sponges and other things.
Did you want to look at those?
Yeah, I wasn't sure if we had moved on, but
we're
yeah,
right
if we could maybe, as long as it's still in our range.
From that front view, it looked like there
Yeah,
was
come
some
on in, Roland.
carnivorous guys.
Yeah, workshop.
While you're doing that, I need to finish answering this chat here about the slug preservation.
Copy. Yeah, just send somebody up.
Yeah, so there's a combination of different
Yeah,
things
just
here.
send somebody up, please.
So
to the control room.
obviously there's this black coral, and then there's these thistle-like things, which have the sort of ladder-like projections across
Nav, do
them.
you know how to
Oh, it's got a nice brill
plot
star
them?
living off
I have
of it.
zero idea.
Me either, so.
Or maybe it's something caught on a carnivorous sponge. That's an interesting
Any more zoom?
idea. Yeah,
Max out.
it's hard to say. But, interesting. And then I think farther, if you pan out further, there's more of these little round, sort of, I guess they're baby Caulophacus or something. I saw a patch of them, I think, on that same rock surface towards the end of the. Yeah, they're to the right there.
Yeah, come on in, Roland.
Yeah, see all these sort of little cute little sponge babies. Yeah, there's like 4 of them in one little patch. Hey, Chris, this might be something you might actually
Video's
look at.
clear.
Oh, it's
Oh.
that.
Oh no, there's all these small sponges, and they're all in the same area. I mean, sort of a kind of a reproductive settlement cluster or something.
Yeah, it certainly could be. It's pretty cool.
Anyway.
Little babies.
Yeah. Anyway, we can move on.
Yeah,
Thanks.
copy that.
Thank you.
So the reason I was silent here is that we've got some recommendations regarding the sea slug preservation, and some various people were weighing in on this. Scott France got us connected to James Aubrey, as well as Megan McCullough, McCullough and also somebody, let's see.
Well, they're talking about Angel Valdez, who's one of the biologists. I think he's at CSU San Luis Obispo. He works on sea slugs.
Yeah, for some reason I thought I saw a message from him, but maybe it wasn't.
No, I don't think he's.
Scott was copying. Yeah. Okay. Oh yeah, from Angel Valdez, right?
Yeah.
So, does everybody agree that you'd like a piece of the foot and ethanol for the 95% sequencing? Because it's not too difficult, and then we can put the rest of the animal in formalin. We do have enough. Is that, are we going to follow? Is that what people would like? What's the consensus here? Because I've never collected one of these animals before, and I certainly don't want to mess this one up.
Should we get a move underway now?
We'll wait for a moment to get a response. Meanwhile,
I'm fingers crossed.
Take a look
USPL's
what's going on the seafloor here.
Say again.
Might have
run out of.
So yes,
there's
you
a
want
lot of these.
You're looking at 326. You still happy with that?
This used to be a fairly active area.
Yeah, 320.
320. Yeah.
Okay, 30 meters.
Is there a waypoint we're heading towards?
Yeah, it's right up here, but we're just trying to get
up on that
higher.
plateau. Yeah. Okay. Yep.
So, okay, I'll get you a 30-meter move at 320. Bridge, RV nav.
Scott's saying it might be relaxed.
It's the bridge, go ahead.
Hey, can we get a ship move, please?
Unless I hear otherwise.
30 meters at 320, 0.2 knots.
Unless I hear
Range,
otherwise,
30 meters,
what
320.
What we're going to try to do is remove a piece
Speed,
of the muscle of the
Good
foot.
copy. Thank
Okay,
you.
now we got a response. Yes, we could chill this down for a bit as well. We do have a freezer aboard, and we can certainly try to chill the animal down to see if we can relax it before preserving it. So how about this? We try to chill it and try to get it nice and cold, and then we will take a piece out of the foot and put it in 95% alcohol. We're also going to take a small piece and put it in the OGL fix.
Video swapping out
for their
what am I
Ocean
looking
up there on the? Is that another?
Are you talking about
and
the tether?
Then the rest
tether,
of it
yes.
we're going to put in formaldehyde, or not formaldehyde, 10% formalin. I mean, excuse me.
How about you keep your heading it where it is?
If
and
that's
I'll push
not
ahead.
acceptable, let me know, because that'll be our plan.
I didn't know what you were talking about.
Yeah. Sorry.
I thought you were pointing at the winch, which
No.
got me very concerned.
Megan says, can we slowly introduce ethanol to reduce the contraction? We haven't so far.
I guess that's all right.
That could be a little bit difficult, but we could try it. What do you think, Matt? How you seeing this? Because we could trade off on this. I'm trying to think how we would do this. So I'm assuming she's suggesting we have like put it in 10% ethanol first, and then 20, 30, etc., and try to do it that way. Okay, so do we have a tentative method here? How we're going to do this? This will. Yeah, what we're going to do, it comes up, and then we can try to photograph it at least initially, as quickly as we can. But on the other hand, we could try to put it in the freezer really quick, because the photographing it is just going to warm it up. That's my only concern. But we can try to get it in the freezer and chill it down, but not freeze it. Right. Yeah, well, I guess it's a matter of whether it's fully contracted or not. If it's contracted up, then we can try that. But then we will remove a piece of the foot in ethanol, remove a very small piece of the foot also to go in OGL fix, and then the rest of it goes into formalin. And in the ethanol, we're going to need to do some changes to that, so we'll mix up some dilutions for the ethanol. Yeah, because that's a small piece, if you see what I mean, so we're going to have to make a bunch of dilutions of the ethanol for that. That's it. Well, that's what they're saying, that introducing the ethanol slowly will do better. No, so we're doing various things. The bulk of the animal is going to go in formalin. It's just straight up in formalin.
Oh, pilot,
you're
can
right.
we take a look at
You're
this
right.
whip in front of us? Thank you.
Absolutely. So Don, if you want to use the Titan, try to keep an eye on the tether here.
Yeah. Okay. Copy.
All right, you're right. I'm getting mixed up here. So you're right.
All right, come on in, video.
So after the formalin fix, then yeah, we'll definitely do the.
It's a partial.
Yeah.
So it looks like we have some kind of bamboo coral here. And we can usually tell that because the skeleton that is below all of this tissue is white with dark nodes, and that gives it the superficial appearance of bamboo. Two of our scientists on shore, Les Watling and Scott France, have done a lot of work with these animals, and maybe they can help us out with which one this might be. It seems to have these very long spines coming out from the polyps. And Scott France declares this to be part of his jade clade of bamboo corals. This is an allusion, or this references a molecular tree that he has, which designates each of these clusters with different letters. So probably
might give us a few minutes to get another ship move in there. If you guys are happy with that. That one just finished.
Thank you, pilot. We're going to move on.
All right, video. When you're good.
All right. We're wide.
Right.
Can we take a quick look at some of those tracks down in the mud?
Where are you looking?
Center, bottom.
Right there?
Yeah, any one of those would be fine.
All right.
So, oftentimes we see sea cucumbers or snails or other kinds of organisms, and they live amongst the sediment here, and they leave tracks that stay here sometimes for quite a long time. Sometimes you can see the animals that make them. Yeah, I think this is fine. I thought maybe we might be able to spot whatever was making the trails.
All right.
Thank you.
Just come on out.
Okay, pilots, we have about 30 minutes left on bottom.
Copy that.
Well, actually, I don't know if you heard, we have 30 minutes left.
Roger that. 30 minutes left. Thank you.
Let's take a look at this.
Yeah, the red coral and the sponge both, I guess.
Yes.
Come on in, video.
This business going on this rock. Oh, there's quite a lot of it going on.
Oh, this is a interesting coral. It's sponge too. Both.
Come and take. I'm going to approach it, so
Okay.
you'll have to adjust focus.
Okay. So this is Hemicorallium, the coral. And the sponge is something that we call Ferrioneroca erecta because it is a form of a species that we have identified tentatively as Ferrioneroca, but it's an erect form, which means it doesn't form a bush. It forms kind of a columnar type of colony. And we haven't seen very many of these so far on the cruise. I'm pretty sure that's what it is. It differs from those leafy sponges that we saw yesterday. And what's that red, bright red thing on the top there? I don't know if you can get a better
some kind of interesting
yeah.
little...
Can you come out a little bit?
Yep. How's that?
Yeah, hold that. I'm going to sit down here, Chris, to get a nice steady shot of that.
Sure, thank you.
Just take a moment. That rock is
I did too,
loose.
sorry. But I'll just go ahead and do it anyway. And I'm keeping time, so then it was Hemicorallium. I'll get that one.
Okay,
We want to
come on
average
in.
0.1 to get to the end of this waypoint.
Copy that.
Yeah, I can't tell from that even. Nothing, there's no details to it. Maybe an amphipod. Or maybe an ostracod, I guess. Not sure. Oh. It's always nice to see brittle stars close up. They have such interesting structural surfaces. This one has, this is a member of the family Okeacanthidae, I think maybe in the genus Ophiopenthaca. It has these very distinctive folds on the disc as well as all of these small, they're either spinules or granules on the disc, and it has a number of arm spines that are quite sharp. Looks like it's crawling over this Hemicorallium. Yeah, I think we're done when video is done.
Yeah, copy that. Just hand down, see if we see anything, and then...
Yes, absolutely. Thank you.
All right. And you're good, video.
Alright, if we can just get kind of a full specimen shot, that would be nice.
Yeah, you want to kill lasers, don't?
Oh, let's take a quick look over to the left there. I think that's another carnivorous sponge.
Oh yeah, I see it now.
Watch, Leaks, do we want to make it up to
Alright, come
waypoint
on
3, or are we okay just spotting these and getting the close-ups of these guys?
So, oh wow, look at that.
I wouldn't mind getting up to waypoint 3 if we could.
So
Okay,
this is
we probably
another
should
nice
moving.
carnivorous
Get
sponge.
moving.
You can see the
Negative pilot, the
projecting
ship has
spines
stopped.
that,
We'll,
Stop the ship moving.
they will
Okay.
hold fast small prey.
Co-pilot, are you
and
still good with 320?
move the items to the center of the
Still
disc.
looks good.
These are actually not possessed of true tissues.
Just
So
3
they
is 0,
actually
0,
form
0
sort
look
of
any better?
unusual kind of stomach-like structures with their cells, which phagocytize, which is to say they use the cells themselves to digest food.
Let me turn a
We
little
caught
bit and
video
see.
of one of these the other day.
Yeah, we're picking up targets over that
capturing
way too.
small
Yeah, you could-
Okay,
portions
I'd like
on
to put
those
that in.
spines.
It's closer to the
Beautiful,
waypoint.
thank you.